think we're going to get started. I wanted to welcome everyone to this, the first guest lecture of our academic year here at Brock University. My name, of course, is Barb Tatarnik, and I'm the manager of outreach for the Cold Climate Oenology and Viticulture Institute here at Brock University. This presentation is being live webcast, so welcome to those online, and I encourage our online participants to input your questions, which we will then pose to our guest speaker here today. The talk will also be archived to the CCOVI website as well for future reference. I'd like to thank everyone for taking time out of your busy schedules to join us with the harvest season just starting. We do appreciate how difficult it is to take time out of your busy schedule, so thank you, thank you, thank you. Peter Salamone. Peter is a technical manager for Laffort, USA. Dr. Salamone has broad experience in industrial biotechnology and the wine industry, both in production as well as wine industry service experience with enological product and enzyme vendors. With this diverse experience, he has become an outstanding technical resource, and we're very pleased to host Peter again here at CCOVI. I'd like to formally thank Laffort USA, as well as our friends from Vines to Vintages, for sponsoring Peter's talk today. And with that, I'd like you all to join me in welcoming Peter Salamone. Peter?
Thanks, Barb, for that nice introduction, and thanks to Debbie and Belinda and the rest of the folks here at CCOVI for having me once again. This is my third lecture here at Brock, and I always enjoy coming here to Ontario. It's always good to visit and see what is going on the other side of the country. I'm based out of California, and I spend most of my time out there, so it's nice to get some diversity of inputs in what's going on in the industry, because things are very different here than they are there. So it's always good to get a different perspective. I usually have a slide that says, who is this guy? And I kind of introduce myself a little bit to give you a bit of rationale as to why I'm up here and you're out there. So I'll just talk about my industry experience for a second. I worked in biotech for a while. My PhD is in genetics and cell biology, so I'm not your usual guy in the wine industry. But I got recruited to a position at E&J Gallo, and we did a number of really interesting research projects while I was there, and that's how I got introduced to the wine industry, and I've been in it for 12 years now. My first year in the wine industry, I watched 1 million tons of fruit go through the crushers in Fresno and Livingston. So I got a indoctrination by fire there. And then I've spent time, I was the lab manager and a product research scientist at Gusmer Enterprises. I've been a technical manager for the Central Coast for Diageo. So I worked for very large wineries. And I was back at an enzyme manufacturer for a while, so on the vendor side as well. So that's just a little bit about how I ended up in my circuitous route. And a little bit over 3 years ago, this position opened up at Laffort, and I was lucky enough to join the team of Laffort. It's a really good team. We're a worldwide global company, and it's got plenty of resources to keep somebody like me interested in this field, and hopefully being able to bring solutions and help drive things forward. So some of the things we think about in Laffort a lot: research, innovation, quality. That's kind of what we're built on. And my talk today is going to have a lot of information and data from different sources. I wanted to list a few of my colleagues that have done some of the work that I'm going to introduce to you today. Some of these folks are from France. Chris Curtin is at the AWRI. Tertius van Westhuizen is our GM and technical manager in Australia and South Africa. So we got a pretty tight global team. We work together quite a bit. So today, of course, my talk is about yeast nutrition, and I've kind of thrown it out there, changing paradigms. So what I'm going to talk about a little bit is what thought processes have been, where they evolved, and where they're going in 3 different areas. One is to talk about yeast and yeast production. I'm going to talk about yeast nutrition and primary fermentation, and I'm going to talk about active dry yeast rehydration and protection. And some of it will be very detailed, some of it will be very applied. So hopefully I'll have enough of both sides to interest everyone in the audience. So always a good place to start. How about the beginning? 6,000 years ago or so is when man is credited with the beginning winemaking back in ancient Macedonia. So, and there's some, these are pictures from California harvest last year, but I thought I'd put them on there anyhow. So, back in the day, what did people think about yeast and making wine? I was thinking about this just in passing, and my mind's wandering, and I'm thinking, they found grapes, people were eating grapes, they said, this is the good fruit, it's a good food source, let's store it. So they probably put it in some containers, and then they went after it later, and it's like, hey, wait a minute, this isn't the same stuff we put in there. And, little by little, they realized, what the product was in there. It used to be good, it was sweet, it was nutritious, and now it's alcohol. I'm sure they noticed that there was a difference when they ingested that. And so they said, this is kind of a good idea, let's try to make this a little more formally. And winemaking's been around a long time. But think about, 6,000 years ago it started and evolved into where, old world, new world, kind of 40, 50 years ago, thought processes on where wines were being made around the world. And had things really changed that much? So somewhere around the mid-1800s, it's 1850 something, I don't have the exact date, but the wine industry in France was experiencing a great deal of problems with spoilage, souring of wines, and there was a preeminent microbiologist in France at the time working in Paris, Louis Pasteur. And the wine industry asked Louis Pasteur to come to Bordeaux and help them with their problems with the spoiling wine because, it had evolved to the point where it had become a big business. They were making large lots. It wasn't just small vats. And it was a real economic driver. Bordeaux was a great trade venue, and wine was a major thing that they moved in and out of there, and the wine was spoiling and having a big problem. Now, Louis Pasteur, the work he did in the wine industry is really important, and sometimes people don't know, but when he disproved the theory of spontaneous generation in the crooked neck flask, that was an experiment he was doing to get at the problems in wine and why it was spoiling. So some of the seminal observations that Louis Pasteur did that made him a very famous scientist came from him volunteering to come down to Bordeaux and work with the wine industry and help those problems. He identified the fact that this organism, yeast, was doing the fermentation, so that's the first time it was recognized that yeast got a name, was doing the fermentation. It was Antoine van Leeuwenhoek years later that would actually look at organisms through his microscope and identify them. But Pasteur is credited with determining that the microbiology was behind fermentation. So it's very important, and just as an aside about Louis Pasteur, he's generally acknowledged in many ways as the greatest scientist that ever lived. His work on immunology and disease, as well as what he did with the wine industry, is credited with saving more human lives than any scientist that's ever lived. So he's a pretty important guy. It's pretty nice that he came and worked on wine. So we learn a little more about it. We begin to understand about the microbiology, some of the chemistry. And another one of the things about yeast, just looking at yeast selection, what yeast's role is just a little bit before World War II and during the initial phases of the World War, there's a fellow named Fleischmann here in the United States that found a way to dry yeast and make it so you could rehydrate it and make it active. And he did that so that our troops in the field could bake bread and eat bread. And there's some old World War II posters about, the bread eaters are the winners. And it's like, it all came from, Mr. Fleischmann and his yeast company, where he developed a drying process where you could transport the yeast and make bread out in the field. So that was, it was a baking innovation at first, but all the other industries that use yeast, it turned that active dry yeast became a real convenience for them, and it was adopted, including winemaking. So what does things like that, innovations like that, really do for you? Before that, it was always just a random act, right? Your yeast were in your winery, or they were on your fruit in the vineyard, and you got what you got in. You had no control over it whatsoever. Sometimes the yeast were strong and would complete the ferments, and sometimes they wouldn't. You had a lot more stuck and sluggish fermentations. You had wines that had a lot more off aromas. This brought a tremendous amount of control to the winemaking process, being able to pitch yeast at a certain pitch rate, population, to ensure that you're going to get a good, strong fermentation and finish. Also, the aesthetic factors of different yeast. All of a sudden now you could grow a single isolate, you could dry it down, you could pitch it into ferments, and you could basically tell that yeast is doing your job. And that's how yeast got isolated from specific chateaus. Most of these isolations of specific winemaking yeast came from the oldest winemaking region, where there was style that was being driven, and that was in France. So a lot of the chateaus that were famous for the wines that they made, that's where the yeast were isolated from for the initial commercial yeast. So you were able to drive specific styles, you're able to drive specific economic outcomes, which is good, clean fermentations. So yeast today. there's an ongoing talk about, do you want to do native ferments or wild ferments? And, how wild are yeast? Are the yeast that we have dried down and we use as active dry yeast, are they really any less wild than yeast that come in from the vineyard or you find in your cellar? I mean, are they more wild? Are they really different? They may be different, but if they were really great at doing what they did, then chances are pretty good that the companies that do these safaris, I know I used to do this when I worked at Gallo, we'd go to all of our facilities and we'd swab all kinds of surfaces and we'd plate it all out and bring it back and we'd pick colonies and we identify all those guys. And then we put them in trial fermentations and we test them out and say, do they have characteristics we want in our wines? And that's how we made decisions on what we choose to go forward. We would grow yeast ourselves based on what we found. But what we found mostly is that the commercial yeasts were better than what we'd find. Okay, so the other point is, wild yeast, native fermentations, these guys were not exactly domesticated, okay? The genetics in the, we've only been drying down these yeasts for, 60 years? And for wine, much less than that. That's a blink of an eye as far as evolutionary timescales. We're not changing these yeasts. They are what they were in the wild. The only way they've been domesticated is that we grow them up in monoculture so we can dry them and be consistent with performance. So, wild versus native. Maybe these things, are they not so wild? Are they domesticated? I don't really think so. But at some points, maybe some of the wild things you want in your fermentations, maybe that's not such a great idea. And I will say, there's a certain amount of complexity that you like to say comes from having a diverse microflora in the wines. Whether you pitch a yeast or not, there's a lot of vineyard yeast and other microorganisms that are going to work and grow in your must up until the time that Saccharomyces generates 4 to 5% ethanol. So even with the best intended consequence, when you inoculate yeast, you're never going to eliminate the wild aspect of fermentations. Those organisms are there and they're going to work until that alcohol gets up to 4 or 5%. So cultured fermentations, where we've gone from putting grapes in a clay pot and seeing what comes out versus having specific strains of yeast that we isolate and grow up, dry down, rehydrate, and pitch to try to control things. What are the benefits of that? Predictable, dependable fermentations. Usually the strains that are isolated are very strong fermenters, so that's one of the reasons. There's always conditions that can happen during fermentation that can impact that. Characteristic production of flavor and aroma. Some yeasts are paired with specific varietals for a reason. They enhance the varietal character of those. And start the fermentation and outcompeting microbial flora. Sure, there's, I don't know how many times I've done this now, but I've done strain identification on finished fermentations. And the fermentation went fine, the kinetics looked just like everything normal, but I pitched the strain in at over 1 million cells a mill, and yet the strain that finished the fermentation is not the strain I put in there. So they're good competitors as far as Saccharomyces goes, but that doesn't mean there's not a good Saccharomyces in there that can still win the race. Many times when I look at finished fermentations and do strain IDs, it's 70-30, 90-10. Sometimes there's 3 or 4 strains in the finished ferment that are identifiable, which means they have to have a certain level of population, usually over 10 to the 4th per mill, or they don't turn up in the tests that we do. So even when you pitch things, it's no guarantee things finish. If you're really concerned about that, I would recommend that you do the microbiology, the strain ID. It costs like $200 to have it done at ETS. Okay. So what else? Complete utilization of sugars. Most of the time you want to ferment to dryness. Sometimes you want to arrest fermentation in specific wines. I get that, but for the most part, when you ferment, you'd like your strain to finish in a nice, reliable way too. You don't want it to get sluggish at the end and finished, but it was about 3 weeks longer than you wanted. That opens a window at the tail end of that fermentation for contamination to occur, or for other yeast that can utilize the sugar that may be there, but in a kind of dormant state just because of population dynamics. It gives them a chance to kick in and start doing things like maybe producing some ethyl acetate or some VA through acetic acid, things of that sort. So you'd like a good strong finish down to dryness. A predictable sugar to alcohol conversion. You kind of like to know, especially, well, I know in the U.S. we have tax laws that change dramatically at 14% alcohol. So many times when you start a fermentation, you'll water back your bricks to make sure that your finished ferment is not going to be over 14% because you don't want to be in that higher tax bracket. There's a certain economics to that. So you'd like to have that be predictable. When you use a strain of Saccharomyces, you'd like to know that if you started at a given brix, you're going to end up in a specific range for alcohol, and it's not going to fluctuate wildly. Good ethanol resistance. Sometimes wines, you don't water them back, you don't mind the tax consequences, you're making a higher value wine, and the alcohol goes over 14%. That's fine. You want a yeast that's going to be able to finish at alcohols that are higher. High SO2 resistance with low hydrogen sulfide production. We're going to talk a lot about sulfur today, sulfur assimilation and its roles. And, these yeasts do vary in their resistance to SO2. Also, I might add that Saccharomyces produces SO2 during the fermentation. So even if you don't put any in there, there's SO2 in your ferment at the end of primary because Saccharomyces will produce it. And low hydrogen sulfide production, I'm going to talk a lot about that later, but of course, nobody likes hydrogen sulfide in their wines. It's the rotten egg aroma, and it's not a pleasant thing. Okay, and also there are other off flavor compounds, or like in terms of pyruvate, a compound that will bind up SO2, lower the effectiveness of your sulfite adds. So you want to produce a minimum of things like acetic acid, acetaldehyde, and pyruvate. And also, you'd like to keep foaming down. That's a factor that can be a property of the yeast strain that you use. And sometimes, good flocculation properties. When the yeast tend to start to fall out of solution and die off, you'd like that lees to be compact. So when you rack off, you have less gross lees to filter to recover wines. Okay, so right in the beginning, I like to make it really clear. Difference between yeast rehydration and yeast nutrition. It's a really big difference, and the products that I talk about that are used for both of these purposes, they're not interchangeable. Okay, we'll go into great details about both of these areas, but I just like to make that point really clear. Rehydration is about survival factors, fatty acids and sterols. Nutrition, you're looking at nitrogen, either from an inorganic or organic source. You're looking at vitamins. You're looking at minerals. Two very different things. And I'll just say, if you use this kind of a product when you're rehydrating your yeast, when the yeast are dried, it changes their membrane structure considerably. And when you rehydrate them, they're like little sponges. They'll soak up everything in their environment. If you put this in their environment, it all goes inside the cell. Cellular metabolism is very complex, okay? And when you just push all this stuff in it, it gums up the works. The yeast don't like it, and they get unhappy metabolically. That's not a good thing. That's when they do things like make hydrogen sulfide or stop the fermentation. So, very important. Know the difference. Okay, in a perfect world, fermentation, you'd like to start off with nice clean grapes, get a good yeast strain, and produce a clean wine, right? And you'd like your yeast to be happy. Happy yeast makes happy winemakers. And so a very important factor in this kind of thing is the proper nutrients and nutrition for your yeast. Some musts contain plenty of nitrogen. Carbon-nitrogen balance is important. I think we all think, or have at least heard about measuring incoming yan. So, but many times, just because your carbon and nitrogen is balanced doesn't mean you have the proper nutrition for that yeast to perform that fermentation through all that sugar and produce all that ethanol to dryness. It's more than just the carbon-nitrogen ratio. Now, in reality, when you start this process, you can have issues. Incoming grapes aren't always perfect. You can have rot issues. You can have enzymatic issues with oxidation, including lactase. And during the fermentation, you can have things like wild yeasts can be in there that can bring detrimental effects. Temperature issues, either heat spikes or drops in temperature. I've had wineries where people are doing, say, a red ferment and someone flips the wrong switch and they chill the tank and it drops the temperature where the people didn't want it to be. Well, the yeast react to these sorts of things, whether it's a heat spike or it's a cold spike. And I'll talk a little bit more about what those things actually do. Inadequate nutrients, mentioned that a couple times already. And so what are the outcomes of these sorts of things? You can have increased VA when your yeast is not happy or when it's experienced a lot of competitive pressure. Saccharomyces can make acetic acid. You're going to raise your VA. Stuck or sluggish fermentations, again, competition, or the epigenetic changes that can occur in the genome of the yeast. It can shut down whole portions of the genome, these temperature spikes. That's how they react to the changes in their environment like that. They change who they are internally and what genes they express. Some of those genes impact their ability to finish fermentations. Browning, this is more an enzymatic effect from some of this sort of issue with the rot up front. Sulfur off notes, again, unhappy yeast tend to spit out bad compounds. We'll talk about that in detail. And then in wines, finished wines, you can have, acetic acid, which is high VA. You can get Brettanomyces contaminations that can lead to 4EP, 4EG and medicinal Band-Aid flavors in wine. So there's lots of pitfalls that can happen. So poor fermentation, there's certainly no shortage of causes. And, low population of viable yeast. Again, the way that works in the fermentation and what's important with pitch rate is that you give your yeast a good head start population-wise. They go through logarithmic growth, and then they get to stationary phase. If they don't have a certain population level at stationary phase, when they start to churn through all that sugar and make that ethanol, they start to die off. The alcohol level goes up. It's a challenging environment for them. It's not what they normally do. Okay, we're asking them to do a job that's not their normal job. We've got to treat them right, give them the right tools. But if the population isn't high initially, when they start to die off, there's not going to be enough at the end when there's still some sugar left, and that's a sluggish and stuck fermentation. Fermentation temperatures, I mentioned that a little bit already and how they can affect the yeast physiologically. Microbial competition, both early and late. I mentioned early competition from, say, vineyard yeast or bacteria that can create things like ethyl acetate and VA, other negative aroma compounds. And late competition, say you've got a bit of a sluggish ferment. The microbial population that's in your wine is never one organism. No matter how much Saccharomyces you pitch in there, and no matter what its growth kinetics are, there's guys that started out in the vineyard and in your winery that are still in that wine. They're in there in the beginning, they're in there in the middle, they're in there at the end. Okay? And if you give them an opportunity where the Saccharomyces dies off, stops growing, there's still some sugar, these guys that have been hanging out in the background, being out-competed, they can kind of kick in their metabolism, and then you can get some late fermentation spoilage. Okay, toxins, these are like microbial toxins. These can be released from some of these organisms that are in your fermentation. Some of these toxins can be detrimental to malolactic fermentation after your primary. Heavy metals, aluminum, lead, these things can come in from different sources. Pesticide residues can come in from the vineyard. So there's all sorts of toxins that can be related to stuck and sluggish ferments. Yeast genetic mechanisms, I'll just say the word prions. I don't have time today to talk about that. But there's certain yeast with a genetic predisposition to turn off their sugar import mechanisms. And that stops the fermentation late, and it's a stuck fermentation. Epigenetic effects, that's where environment will change the expression patterns in the genome. I mentioned that in response to temperature spikes. And nutrition. I'll spend most of my time today talking about fermentation issues related to nutrition, including carbon-nitrogen balance, Brix and Yan, I've mentioned that, macronutrient shortage, micronutrient shortage, and trace nutrient shortages. These are all factors that are important, especially when you think about, we're asking the yeast to do a job that's not really what they do. Yeast exists just to reproduce and make more population. It's not in their best interest to sit in stationary phase and spit out a bunch of ethanol. It doesn't do them any good. But we harness these guys and we bring them in and say, we want you to work for us. We're going to put you in this environment, and here's what we want you to do at the end. And we know you can do it, but you don't like to do it, but we're going to give you everything we can to help you do it. And that's where the nutritional aspects come in. Carbon-nitrogen ratios are very important, and that's kind of one of the paradigms. From not knowing a lot about things and not knowing how to manage things to learning about measuring Yan and incoming must, matching it up to Brix levels, and then supplementing your Yan to generate a nitrogen-carbon balance that's going to enable the yeast to complete the entire ferment in the way that you'd like. Okay, so but now carbon-nitrogen is important, and that was, you can see the tables for Brix and Yan additions, and that was developed, I want to say, somewhere in the 1980s. I was looking for that seminal paper, and I couldn't find it. I only found a 1990 paper out of the Bassan lab where they talked about this, but I'm pretty sure the work was done when Christian Butsky was still in Linda's lab, and they developed that initial table. I'll talk a little bit more about that in depth later, but that's important, but it doesn't tell the whole story. Just carbon and just nitrogen won't get the yeast through. There's a lot of micronutrients that are important, and we don't measure micronutrients in incoming yeast. And really it would be counterproductive from a time standpoint, from a resource standpoint. You're much better off to understand that the yeast need it, and when you do your nutritional adds, take that into consideration. Get some organic nutrient in there. Use a complex nutrient or use an organic nutrient so that you bring some of these trace nutrients into play. Okay, so I mentioned happy yeast, though that leads to the point that some of them aren't happy, which would make them unhappy. What's some of the consequence of you making your yeast unhappy? Stuck and sluggish ferments, hydrogen sulfide, another aroma consequence. Our friend Saccharomyces will do this to us if we don't keep them happy. Elevated C8 and C10 fatty acids. What's that all about? Sometimes late in fermentation, if Saccharomyces is a little bit unhappy, it gets finicky. It's like a toddler that's hungry, okay? They throw tantrums. Well, when Saccharomyces throws a tantrum, it'll spit out these C8 and C10 fatty acids into its environment, okay? And you say, well, who cares? What's that? That doesn't matter to me. These molecules are inhibitory to Saccharomyces. It's trying to carve out its own niche. It's being limited nutritionally, and it's hungry, and it's trying to carve out its own environmental niche so that it can get what nutrients are there. Okay? Now this, at higher levels, C8 and C10 can be inhibitory to Saccharomyces, but at lower levels, at 25 mg per liter, it's inhibitory to Enococcus. So this is a guy that can affect your malolactic later. So I'll say this, if you've got a malolactic that's really sluggish or you can't get it started, think back to your primary fermentation. Was it sluggish? Did it have any problems? If you had problems, this may be your issue. Okay? Now the bad news is that this can cause problems. The good news is that you can remediate it. Okay? C8 and C10 fatty acids, if you fine your wine with yeast hulls, okay, the yeast hulls will absorb these fatty acids onto their surface and they'll sediment. So you can detoxify your must if this is your problem. If you've got a stuck malolactic, one of the first things to do is run a detox and then try a restart. Okay? You don't have to measure C8 and C10s to see, I mean, you can. I've worked with ETS to develop this. I did a survey across California about C8, C10 fatty acids in wines. I asked Gordon at ETS to develop an assay, and he did. And it's not in their website, but if you ask him to run this assay, he will. So if you want to know if you've got over 25 parts of these, there's a lab you can send a sample to get it. But in reality, the symptoms of this are stuck malolactic. So you can just detox and repitch. If that doesn't work, then you've got to start trying to do some more remediation. High SO2 production by the yeast. This again, you can stress your yeast, they make SO2, and that can become inhibitory to finishing your fermentations. Restarts are much more expensive than prophylactic nutrient adds. You know, this is kind of old news. This is an old paradigm. It's been, 20 years since nutrients were introduced, nutrients beyond DAP, okay, where we started getting complex nutrients, yeast extract blends with diammonium phosphate. And there's plenty of work out there. We've got a table that we've published at Laffort about how much it costs to do a restart. And the percentage of stuck fermentations that existed before nutritional interventions came in. And it costs more to do the restarts than it does to just use nutrients across all of your fermentations. All right, negative impact on subsequent malolactic. We've talked about that a few times already in specifics, including C8 and C10s. So, nutrition. Basically, I've already mentioned macro, micro, and trace nutrients, so I'll just go through this really quick. Macronutrients, CHOMPs, C-H-O-M-P-S, I learned that basic microbial physiology. We add potassium and magnesium as macronutrients in wine fermentations. It's important for Saccharomyces in the must environment to have these ions in fairly high concentrations. Micronutrients, things like vitamins, the B-complex vitamins. Here's a list of the 8 major B vitamins. Minerals, including these major minerals. I've got a full slide. I'm going to talk about thymine in detail, but I'll just say real quick, vitamins and minerals, what is their real purpose? What do they do functionally? Why do these cells need these things? These are cofactors to enzymes. Okay? Enzymes are each step through every metabolic pathway in the organism. Everything they do metabolically, producing amino acids, producing lipids, making more complex molecules, everything these guys do is mediated by an enzyme. And a good deal of those enzymes have cofactors at their active site. The catalysis that they're there to do is dependent upon these cofactors in those enzymes. Without these, metabolic pathways get extremely stressed. When you stress a metabolic pathway, it backs things up. When things get backed up, yeast get unhappy. Yeast get unhappy, winemakers get unhappy. So, all these factors are important, but supplement additions are usually based on YAN and Brix. You add a certain amount of nitrogen based on what your incoming YAN is and what your Brix are and trying to balance that. Does that necessarily balance everything else? Balance your YAN and it balances your carbon, but not necessarily all these micronutrients. So, when I finish today, I've got a very valuable prize. You know, we'll have a test about this diagram afterwards. The winner will get the prize. No, just kidding. I like to throw this up there just to illustrate the complexity of metabolic networks that I was just talking about. Every one of these reactions, this flow chart, here's your basic glycolysis, here's your TCA cycle, amino acid biosynthetic pathways, lipid biosynthetic pathways, all this stuff's going on inside the cell, and this is just a portion of it. This is how complex it is in there. This is what is life. In a closed catalytic system that is a single-celled organism, this is what's going on in there. And every one of these steps is mediated by an enzyme that needs those micronutrients to function properly. Okay? It's complex, and it's extremely interrelated. It's one of the reasons why you take like something very non-specific like a yeast extract and you feed it. Nobody wants to figure out where the deficiencies are in each little thing here. You'd like to have a blanket cure that you can give and say, let's make this machine run. Okay, and that's kind of what those complex nutrients do. Okay, I said I was going to talk about thiamine, and I am real quick. Thiamine, vitamin B1. Why is it so important to us? Okay, we make wine, we employ Saccharomyces, we feed it grape must. Why is thiamine important? Okay, first of all, it's a pretty complex molecule. Okay, this molecule to create from scratch in the yeast takes, I'm not going to go through every step, but there's a number of biochemical steps, each mediated by a different enzyme to produce this molecule that is then used as a cofactor in another enzyme. Okay, that's a big metabolic burden. There's lots of molecules like this. Each of the B vitamins, yeast can biosynthesize their own vitamins, but there's a metabolic cost to that to them. These are complex, and it takes a lot of steps and a lot of energy and resource on their part to make these. Same with sterols, same with fatty acids. So when you supply them things like vitamins and sterols and fatty acids, they've got internal regulatory mechanisms. They will shut down the biosynthetic pathways because they already have these molecules. It's endpoint feedback, okay? And it saves them a lot of metabolic energy, and it reduces their stress in the fermentation environment, okay? So this is thiamine, and it's important. Well, let's talk about where it works. You can see this is the enzyme that thiamine works with. This little piece right here, that's the thiamine pyrophosphate right there. That's the cofactor at the active site. This is a monomer in the enzyme complex that is a tetramer. So this is the actual enzyme. This is 1/4 of this enzyme. That's the thiamine right there. But it's critical for this because this is the enzyme that does the catalysis from pyruvate to acetaldehyde, which is subsequently reduced to ethanol. Okay? Now this is the reaction. This carbon is going to flow through the glycolytic pathway and you're going to end up with pyruvate. Okay? But because it's anaerobic, you're not going to push this through the TCA cycle. This isn't going to, it's not going anywhere unless you proceed with this reaction. This pyruvate decarboxylase, this is this enzyme. So without a lot of this enzyme and in turn a lot of this cofactor, this reaction to ethanol is not going to proceed. You've put your yeast in this environment that's high carbon, and there's no oxygen, there's nowhere to shunt this stuff. This is what you're asking them to do. So you've got to give them these tools to do the job. Okay, I mentioned the Brix and Yan table, and again, just for the sake of, I'm trying to keep in line with the whole shifting paradigms thing. I want to talk about this just for a second. You can see, here's a usual thing, amount of Brix and the recommended amount of Yan. These tables that were made up and get perpetuated now, these things really helped alleviate a lot of stuck and sluggish fermentations. Knowing that nitrogen was important, that it was deficient in a number of musts, this was really important stuff when it came out. Okay? It helped move fermentations from a real risky area into a much more manageable area. A lot less spoiled wine, a lot less remediation. Okay? But when this was done with a fairly inefficient yeast. Okay? And it was done, the Yan supplementation was strictly with DAP. Okay, yeast strains, we've evolved in the strains we use. We've isolated strains that are specifically more efficient in their utilization of nitrogen, and also with the characteristic aromatics that they bring, which puts a metabolic burden on them as well. And we've also changed with the Yan supplements that we've used. We use complex nutrients and even organic nutrients today that are used at different efficiency ratios than DAP. So why this was really good when it came out and worked for a number of years, and it's still valid to a certain extent, maybe we know a little bit more today. So I just like to show this. This is actually a table where you've got, this is Yan, parts of nitrogen. Each one of these colored bars represents a different year, 06, 07, 08, 09 through. And these are different vineyards, Chardonnay 1, Chardonnay 4. These are vineyards in Pennsylvania. Okay, so now that you know the axes, here's the different levels. This is a low Yan at 125 parts, and here's a higher Yan at 225. you can see that year by year and vineyard to vineyard, these yans change. So my point with showing this slide is measure your incoming yans, please. Even if you think you know your vineyard, even if you think you know how the season went, measure your incoming yans, because you don't really know where they are until you measure them. And, like I like to say, you can't manage what you can't measure. Okay, so I talked about this, making the yeast happy, and this is just a bit of all of these kinds of sulfides that the yeast will produce in response to. See, when you gum up their metabolic networks, what happens is they still assimilate sulfur, but they got to dump it somewhere. If they don't have the right carbon skeleton, cysteine or methionine, some of the productive ways for them to store sulfur, then they start putting it on other molecules, or they start excreting it as hydrogen sulfide. This is just a list of a lot of these things. And just to say, these are called light sulfur compounds, and these are called heavy. These are more your disulfides. So even if you're not smelling hydrogen sulfide, your yeast, if it's stressed metabolically, can still be producing these guys that have a much higher threshold of detection. So these can be in there, you may not smell them. But once you put your wine in a bottle and it gets in a reductive environment, these disulfides can split and become mercaptans. And mercaptans have a much lower threshold. So all of a sudden you say, oh, my wine wasn't stinky during the fermentation. Why does it stink like hell in a bottle? It's because your yeast really was unhappy. You didn't know it at the time. These things don't get formed in the bottle later. They just get revealed later. Okay, so I talked about Archaea represents our friend Saccharomyces cerevisiae with its little offspring bud over here. And so really, as a real simple way to look at this, you've got sulfates coming in, you've got a nitrogen pool and amino acids being imported into the cell. You've got these pantothenic acid being a very important cofactor in the production of these carbon skeletons for the sulfites to get attached to produce positive sulfur compounds. Okay, this is the pathway, the sulfur assimilation pathway, along with the carbon pathways that develop the skeletons that this sulfur gets attached to in a positive way metabolically. Okay, that's what can get off kilter. When it gets off, either due to low nitrogen or a lack of another one of the B vitamins, pantothenic acid. Okay, these two things have been shown to greatly impact the sulfur metabolism and the development of off flavors. And like I mentioned, shortage of nitrogen can do that, and shortage of pantothenic acid. Oops, wrong button. Okay, so, and here's some work that was done. This was done, I want to say, okay, I'm not going to say. I thought I had a reference down here. Alrighty, but real quick, this is total hydrogen sulfide on this axis, and this is pantothenic acid. This is in a very low yan fermentation. This is a fermentation with only 60 parts of yan. Okay, so we stressed it on purpose. And then we threw only 10 parts of pantothenic acid in, or 10 micrograms per liter, it's less than a part. And look at the level of hydrogen sulfide, huge. Okay, low yan, low pantothenic acid, lots of hydrogen sulfide. Bump the pantothenic acid up to 50, cut this down by a third. Put it up to 250, cut it down even farther. Okay, which goes to show pantothenic acid has a big impact on hydrogen sulfide production and sulfur metabolism, but with a super low yan, you can't completely overcome that and eliminate it. So what happens if we put this in a high yan environment, okay, and we put the same levels of pantothenic acid? You can see that with low pantothenic acid at a higher yan, you get even more hydrogen sulfide produced than you did over here. But once you start to balance it a little, get the pantothenic acid up to 50, you can cut it down, and then you can make it the lowest of all with 250 pantothenic and 250 of yan. So it's an interaction between these two components, which goes to show it's not just nitrogen supplementation that can keep your yeast from producing hydrogen sulfide. You need to have pantothenic acid, vitamin B6 in a certain amount as well. And again, nobody measures pantothenic acid, and it's actually, I'm sorry to say, I don't know the regulations from Food Canada on pantothenic acid additions to wine fermentations, but in the U.S., only thiamine is approved as a supplement. You can add pantothenic acid if you add it as yeast extract, because there's plenty of these vitamins in yeast, but you can't add pantothenic acid as a purified molecule in the U.S. I already do that. Okay, so managing hydrogen sulfide formation, just some quick guidelines. Choose the correct yeast strain. Some are more prone to this than others. Know your wine style, know your varietal, okay? There's certain wines that are certain musts that are more susceptible to this. They have more sulfur compounds in them, so the yeast may make more of these compounds. You can get yeast strains that have lower propensity to make sulfides. So again, know your yeast needs and characteristics. Needs. Yeast come with different nitrogen needs. There's ones you got to feed more to than others, and your vendor should be able to tell you that. Measure the YAN and adjust accordingly. There's nutritional strategies. Adjusting doesn't mean just throwing DAP at it. It doesn't mean just throwing an organic nutrient at it. If you've got a really low YAN, it's good to come with some DAP to build that up and then finish it with some complex or organic nutrients so that you build that micronutrient into it as well. Pantothenic levels, I say make sure the pantothenic levels are in balance. How do you do that? I don't even know of a nutrient that measures pantothenic acid and says we have this much. So really, the best advice I can give you on that is don't just adjust your YANs with DAP. Use a complex nutrient if you've got pretty good nitrogen coming in, or if you've got really low nitrogen. Generally, you have low everything if you have low nitrogen. So you got to put a portion of organic nitrogen in there so you get more of those micronutrients. And another thing that can work greatly in your favor in terms of production of hydrogen sulfide and sulfides is the turbidity of the must. Some yeast like 150 to 300 NTUs, and some of them want to be 50 to 100 NTUs. And that's, again, your yeast vendor should be able to talk to you about what the solids levels that are optimum for the yeast. We look at these things when we develop them, so should be able to talk to you about that. All right, and maintain good viability through fermentation. Again, we talked about viability. It can mean a lot of different things, but really, if your nutritional status is good from the beginning, then you're going to get a good, strong exponential growth curve. You're going to have a good population. And then during some, of course, red wine fermentations, aeration can help these yeast, especially during, I'll just say this real quick without doing a lot of metabolism, but during exponential growth, okay, the yeast are using a lot of energy, okay, and they get 38 moles of ATP from a single molecule of glucose aerobically working that molecule through their system. They only get 2 net anaerobically. So some oxygen during early fermentation in reds really helps those yeasts get robust and to be strong. Okay, like I mentioned, this is just a list of Laffort yeasts, and this is nitrogen need, and you can see that with our yeast we have low, average, and high need yeast. This is basically twice as much nitrogen need as these guys. So there's a difference, and it can make a difference in how you approach your nutritional strategy for your fermentations. What's your varietal, what's your incoming YAN, what's its history in terms of fermentation and odors, and also positive aromas. Choose your yeast base to supplement your positive aromas, and also make sure you feed it right so that it creates less negatives. All right, and again, inorganic nitrogen versus organic nitrogen and utilization curves. Real quick, this is just, your YAN. The dotted line is a sugar curve. This is your sugar depletion curve of a nice normal fermentation. It doesn't go straight to the finish. It's not dry in a linear fashion. It curves off a little, but it's a good, strong finish. And what happens in relation to that sugar utilization, your nitrogen utilization with amino acid nitrogen or organic nitrogen, it's a moderate curve. It kind of follows the sugar curve, and then the amino acids will increase over here as the yeast go through autolysis. Okay? But with DAP or with just mineral nitrogen, the yeast take it up very rapidly. It's a hotter fermentation, and then there's no more in the ferment. And this is very much similar to like feeding your children candy versus giving them a good balanced diet and then watch their behavior in a closed room. Okay, so what are the benefits of a better managed fermentation with a more moderate uptake of nitrogen, more moderate sugar utilization? Slower rise in temperature can allow for better extraction in red fermentations, less foaming issues. It's not as vigorous with the CO2 evolution, less hydrogen sulfide produced. Yeast, again, yeast like moderation. If you don't push them too hard in any one direction, they're pretty happy and they'll do everything you want them to do. Clean fermentation completion, a nice finish without being sluggish, reducing the risk of contamination and off aroma generation, and flavor aroma development. If your yeast are happy in a modern environment, okay, you want them to produce some fermentation esters or reveal some thiols or generate some thiols, all these flavor aroma compounds that are characteristic of the yeast. If their metabolism is stressed and they're really busy just trying to earn a living, they're not going to make all that extra stuff for you. Because that's not their primary thing. They'll do that as like a secondary metabolism. But they've got to be happy in their primary metabolism first. Getting the proper nutrients in place will allow them to do that. Like you can get the best yeast in the world. If you don't feed it right and the fermentation isn't strong, it's not going to perform. Okay, real quick, I've talked about these different nutrients, and I've mentioned mineral nutrients and complex and organic, so I just wanted to illustrate that a little. I've talked about micronutrients and macronutrients, okay? Our products, just as an example, there's DAP alone, or inorganic nitrogen. We supplement our DAP with thiamine, because even if you're just using DAP, I've expressed the importance of thiamine. We think it's important enough to make sure that it's in this product. So you've got this kind of a product that's a mineral nitrogen with a little bit of supplement. So there's a little micronutrition in the form of the thiamine, but there's a lot of macronutrition in the form of a lot of nitrogen. Then we've got what we call complex nutrients, and this is a blend. It's a yeast-derived component along with DAP. So you've got some macronutrition, you've got a pretty good deal of nitrogen in here, but you've also started to add more micronutrients in the form of the yeast extract. And then this other product category is organic nitrogen, and what's in here is it's basically all yeast extract components. So you've got a lot of amino acid nitrogen, there's a little ammonia, because when you pull out of yeast, there's ammonia there. So that's very heavy in the micronutrients and very low in macronutrients. So and what you would want to do is if your YAN, the initial YAN is really low, okay, you want to use something like this to bring it up to a moderate level, and then you want to finish it with something like this to make sure your micronutrients. So you do a combinatorial approach in instances like that. If you've got a medium YAN and you're comfortable because you do some other techniques, maybe a good rehydration where you lower the nitrogen demand, you could strictly go with an organic nutrient. If you're very close to where you want to be, or even if you're where you're at in terms of YAN, you might want to think about maybe a third of the way through the ferment, throw your yeast some organic nutrient, give them some of those micronutrients after they've depleted things that are in the must. So even if you have good YANs coming in, you still might consider a nutritional strategy that's not a mid ferment, but a first third ferment add. Okay, so I'm going to change gears here. I've done nutrition so far, and I mentioned rehydration, but I'm going to focus on that pretty much for the remainder of the talk. And yeast survival factors, this is what the rehydration is really all about. Again, I mentioned it's not the nutritional products. Please don't use nutritional products to rehydrate your yeast. You won't be happy. Your yeast won't be happy. Okay, so basically what these things are, what's really important, fatty acids and sterols. Okay, there's other components in these, but what they're really doing, what they're enriched in, when we produce the product, we enrich it in these components, and I'll show you exactly what they're doing for the yeast and why it's important that when you rehydrate your yeast, this is one of these paradigm shifts. Okay, it used to be you used the yeast that came in on the grapes. Then it was you had an option to pitch the yeast and get the population up and focus which one you wanted to use. But there was a price with that. When you just use warm water and then acclimation with must, it's a different thing than if the yeast would come back and they'd be viable, but it wasn't 100% viability return, and they weren't metabolically as active as they could be. And what we learned through doing processes like this, experimenting with rehydration products before we make them available commercially, is can we help this yeast be more active metabolically early in the fermentation? Okay, so start with some basics, some nice diagrams. I always like pictures. What we're talking about with the rehydration products is when we're taking an active dry yeast and we're rehydrating it and bringing it back to life and wanting it to jump right in and get active metabolically and work hard for us, what we've done with the drying process is we've affected the structures in their outer membrane. And you can see, like in yeast, they've got an outer cell wall that's got a bunch of interesting polymers in it, as well as these this is a protein. This purple glob represents a protein, and this represents like a high mannose chain. Okay? And then you've got a space in between here where there's enzymes and other proteins, structural proteins, as well as some carbohydrate polymers. And then you've got your lipid membrane, your lipid bilayer with some embedded proteins in it, in the membrane. So if you take a closer look at this membrane, okay, when the yeast get dried down, you pull all the water away from these surfaces out here, it can change the way these molecules interact with each other. And these are fatty acids, these gray strings here. And what can happen with those fatty acids is they can get intercalated like this. And that changes the dimensions on this membrane. The distance from this polar head group to this polar head group is shortened. Okay? And what that does is it impacts the way that these fatty acids and polar head groups interact at the surface, the transmembrane surface of these proteins. Okay? These proteins' function is essential that these be associated in the proper way. When the yeast get dried down, it's not in the best conformation. The good news is we can bring that back. If it's been dried down and these things get intercalated and they're kind of sticky with each other, it makes the whole thing kind of stiff. And the way to make that loose again is to create some fluidity in the membrane. That's what these fellows do, the sterols. These sterols are these ring structures, these yellow and green guys here that we've got illustrated. What they do is they provide fluidity in the membrane. Okay, and the way that works is kind of like, think about a stick of butter in the refrigerator and a stick of butter out on your countertop in the summertime. Okay, the difference in those textures is really the difference between sterols with these fatty acids and without them. Okay, so if you can loosen this up, loosen that whole membrane up, the bilayer, then it can associate properly with these transporters and everything can function appropriately. So, and again, these are things that we've learned through applications, but also published by, Landolfo et al. Is the seminal paper about oleic acid, fatty acids, and sterols, and specifically ergosterol, being important for minimizing oxidative stress, which means basically better ethanol resistance. Okay, and that's one of these things that the rehydration products do. There's really two major functions they do. They provide good fermentation finishes, resistance to stressful environments at the end of fermentation, specifically ethanol and oxidative stress, and they help the aromatic profile. They will allow the yeast to produce more positive aromatics and reduce the negative aromatics. Okay, we'll show that as we go through. But what this paper showed is through lipid and sterol supplementation, it reduced the biomarkers of oxidative stress, including resistance to ethanol and superoxide dismutase activity, which is, again, stress mechanism in the organisms. Increased cellular antioxidant response, superoxide dismutase. Increased cell viabilities, initial viabilities. Upon rehydration with this product versus just warm water, you can measure how many live cells are there and increase the viability. And reduce production of acetic acid. Like I said, this acetic acid production is strictly about the stressed metabolism of Saccharomyces. Reduce the stress, reduce acetic acid production. Okay, so I mentioned this about these. What I like to talk about with this particular slide, it's not as pretty a diagram, but you can see it's the same thing, is to talk a little bit about these transporters and why it's important that these are in the proper structure and that they function appropriately when the yeast rehydrated and put into the must. These guys are responsible for bringing in ammonia, for bringing in amino acids, for bringing in glucose, and bringing in everything else that's in the environment. They have specific receptors on the cell surface that bind these molecules in their environment, and if they're there, they'll bring them inside. But there's also a lot of regulatory mechanisms inside that will shut these things down, okay, when they have enough. Which is why I say don't put nutritional products in your rehydration water, because what you do is you force all of this stuff inside and it's out of balance. What the yeast wants internally for its metabolism is balance. So I like to show this slide because I like to point out, we've seen these little cutaways with a couple of little globs on it. This still doesn't do it justice, but each one of these colorful things on the external ring here represents a transport protein in the cell membrane, okay? And also this basic metabolic pathway and some arrows here represent a lot of the regulation, the communication that exists inside the cell from what it senses in its environment through these transmembrane proteins and how it's regulated, whether it brings it in, whether it doesn't bring it in, and also the signals that go back and forth in terms of regulating metabolism, all the way into regulating inside the nucleus the gene expression. Okay? It's very complex. There's lots of these things. It's not a simple one-on-one. Okay, so just to show some of the data that illustrates sterols and viability, this is a particular study that we did at Laffort, and you can see these are viable cells on this axis. So you can see that we started off with a pitch rate and viable cells grew very rapidly, logarithmic growth phase. These first two lines, these pink lines, represent a 25 degree C culture. Okay? And you can see that in C1, where there was no ergosterol addition, that the death rate was this particular slope of this solid line. But with the sterol addition, ergosterol, the death rate was reduced. Now, at an elevated temperature, 35, you can see the death rate without ergosterol was extremely steep, and when you added that sterol, even at an elevated temperature, it reduced the rate of death. So, just again, experiment to show this isn't just, we're not making these words up and saying that they do stuff. All right, so I mentioned the fermentation and good strong ferments and cell viability. The second half of that is more aesthetic when you use these products. I said more positives and less negatives. So again, we've done a lot of work on this. We developed the methods to measure these thiol components, these flavor aroma components. This is in a Viognier, for instance. This is the amount of these compounds. This is 4-MMP, which is the boxwood or grassy character 3MH is grapefruit citrus character, and this is passion fruit, 3MHA. You can see the white bars represent a fermentation without the rehydration product, and the gray bars represent with. You can see in the Viognier, there's not a lot of MMP to be revealed. So again, you don't see much of an increase, but in the fruit character of the citrus and the passion fruit, it's much elevated, much more characteristic flavors of Viognier. I don't particularly have apricot on here, because we don't have, I don't think we have the assay for the particular molecules that are apricot. But you can see that in this variety, you get an increase in the specific varietal flavor aroma compounds. Here's an example from a Sauvignon Blanc, and you can see in Sauv Blanc there's a lot more precursors of MMP, so you can see a bigger increase there, but also an increase in the grapefruit and the passion fruit flavors as well. So varietal characteristics of the thiol-driven flavor aromas with rehydration versus without. Also, I said more positives, I said less negatives. Let's take a look at on this particular experiment. This is kind of interesting. It's a Cabernet Sauvignon fermentation, no DAP or organic nutrients added. It came in at 150 parts YAM, so it's a low YAM, and that's good. We intentionally stressed this. Okay, we stressed this fermentation, and then we either just did the fermentation or we rehydrated the yeast in Dynastart first. You can see that without any rehydration in this stressed environment, it produced this much hydrogen sulfide and this much of these other rotten egg, stinky sulfide aromas. But just with the rehydration, with no additional YAN, no more nitrogen, look at the reduction in hydrogen sulfide and these other stinky compounds. Less negatives. So this is a real nice piece of work that we did at Laffort, and I want to take a minute just to go through this. This is a paper that we published. Gail Winter was the lead scientist. She was at AWRI working in Chris Curtin's lab when she did this piece of work. Tertius is, again, our director down in Australia. He was an advisor throughout this work and actually, suggested it. So, and what this is without looking at the title, it's the use of transcriptomics to investigate gene expression and metabolic stress in response to lipid and sterol addition at yeast rehydration. So the cool thing about this is that it looks globally, all those diagrams that I put up before, that's kind of like, years and years and years of really good biochemists doing a lot of work to decipher exactly what biochemical pathways they are. But what we have at our hands today with some of the molecular tools we have, we can actually look at those pathways in action and quantitate them. And that's what we did through the use of transcriptomics in this work. So real quick, central dogma of molecular biology. You've got DNA being transcribed. Well, let's start beginning. Replication. DNA gets replicated. That's how we make more life. Cells replicate their DNA. We replicate our DNA and divide. That's the first part. But DNA can also be transcribed into RNA. Okay? And there's different kinds of RNA, but for what we're talking about today, let's talk just about RNA that encodes for genes. Okay? So messenger RNA. The RNA gets made, it's a copy of the gene that's in the DNA, then the RNA gets translated into protein on a ribosome out in the cytosol. These events occur in the nucleus in a eukaryotic cell. This event occurs out in the cytoplasm for the most part in the eukaryotic cell, and you make a protein. So here's your central dogma, right? So when you're looking at proteins, remember proteins are each step in a metabolic pathway. Those conversions of those metabolites are all mediated by enzymes, which are proteins. Okay? Where does the proteins come from? Messenger RNA is translated. Where does this come from? It's transcribed from genes. Okay? And this, like I say, in location, it represents a nucleus inside a cell, and you can see replication occurs here, transcription occurs here, mRNA gets transported out of the nucleus and translated into proteins. So how does that work with what we do with the transcriptomics study? What you do is, well, how this works is you'll make a bunch of DNA clones of, say, the genome of an organism, and then you'll do PCR amplification of specific known gene sequences, and through robotic printing on a borosilicate or glass surface, you'll embed these specific sequences in a grid on this piece of glass. Thousands and thousands of these. This is your basic tool. That's how you make your tool. And this is what we've done with the yeast genome. We've got the yeast genome right here, right there. Very easy. Okay, so, and then you'll do like a test and a reference. What you'll do is you'll harvest some cells at a certain stage of metabolic activity. And then you'll harvest all the messenger RNA from that. And then through the use of a reverse transcriptase enzyme, you can make the coding DNA for that messenger RNA. Then you'll take that DNA that you made through reverse transcription and expose it to this slide, and it'll find its partner. Okay? Now these things are labeled. You label these guys when you make them, and then you can excite these labels with, say, different laser wavelengths, and then it'll glow, and you can read where those genes hybridized, and you can then tell which genes are being actively transcribed and in what amount. So this is a chart from the, and I'm sorry for the bad copying here, but it's not really that important to know which of these things are. This is a bit of the data from the paper that we did with that transcriptomic study. And you can see this is a, MIPS is a database that provides functional identification of genes that have been annotated, okay? So these are MIPS functional categories. Each one of these blurry things that you can't read is a different gene-related category. And then so you can see that this blue bar, this blue bracket, is amino acid metabolism genes, and this blue bar is sulfur metabolism, which overlaps with some amino acid, which makes total sense. Methionine is a sulfur-containing amino acid. Cysteine is a sulfur-containing amino acid. These pathways should overlap, and they do. And you can see now when we go over here and look at the data, the green versus red bars, the red represents upregulation, and the green represents downregulation. Now, what do you think is stressed? If this pathway is stressed, if these gene products are stressed, it's upregulated. The organism's not getting enough of that transcript. It's being limited in that metabolic pathway, and it's saying, make more, make more. Okay, so it's stressed. All of these are not stressed. This is a response to the Dynastart rehydration. These pathways, the sulfur assimilation pathway and the amino acid pathway, stress relieved. So this is kind of even blurry. This is just a blow-up of the sulfur assimilation pathway, okay? And again, this is a specific response through some of these, and you can see that N, these green, or the one on the left is an N. I thought it was important for me to go through and fix this blurriness just with these specific areas. The N represents the Nutristar addition, and the D is a DAP-only addition. Okay, and the response is, again, red means upregulated and green means downregulated. Down means it's not stressed, red means it's stressed. You can see that with the did I say Nutristar? Okay, N is, it's a Dynastart, whatever, it's a rehydration product. You can see that it reduces the stress in each of these green boxes, in each of these categories, throughout this entire sulfur assimilation pathway. And if you do DAP alone, nitrogen alone to relieve this, it doesn't really relieve that much stress here. So the nitrogen relief is more in the amino acid pathway, not in the sulfur assimilation pathway. So think about it. Every amino acid has an amino group. It's got a nitro group on it. Not all of these guys have nitrogen. So the way that nitrogen works to relieve the stress on hydrogen sulfide production is by allowing amino acids to feed in here and accept that sulfur and become methionine and cysteine instead of other carbon skeletons that lead to sulfides that stink or a lack of proper carbon skeletons and the organism secreting hydrogen sulfide. So again, this is a chart, again, a metabolic chart based off of that gene chip, okay, for the transcriptomic study. The genes that were embedded on that chip represent these different dots. Each of these dots represents an enzyme in a metabolic pathway. And here's a map of how these are related in different pathways in the yeast. Okay? Now in this experiment, we used a Riesling, we used the yeast VL3. The YAN was only at 120, and it was about a 21 Brix, pH 2.9. These things aren't really important for this experiment. But you can see that, okay, with DAP addition up to 250, so we added 130 parts of DAP. And then we took a look after we extracted the RNA after approximately 15 grams per liter of sugars. Okay, we pulled that out after the yeast started metabolizing, and we took a look and said, what's going on transcription-wise inside this yeast? You can see this is the sulfur assimilation pathway. This guy is stressed out. Look at all the red. Look at the red around the rest of this whole thing. This is DAP addition alone. So some of the amino acid pathways aren't stressed, but there's a lot of other stresses in this organism still. This guy is not a happy cat. He's a stressed cat. So if we take the exact same experiment, exact same must, same yeast, same incoming YAN, same everything, with no DAP addition whatsoever, but we rehydrate the yeast with Dynastart and then do the fermentation, pull the yeast at the exact same time in the fermentation, very few red lines, sulfur assimilation pathway, stress relieved, fat and happy cat. This guy's good to go. So that's really the take-home message about that particular study. You know, we're really looking in a global way what's going on inside the cells and trying to relieve as much metabolic stress as possible. Okay. All right, so really, and then I just want to, I got to throw this out there. Every rehydration product that's out there is not the same. Every nutrient that's out there is not the same. Okay? The point is, and we've shown that sterols are very important to provide the benefits. This is, and again, I'll say, this isn't the only way that yeast can get sterols and increase the sterols in their membranes. Yeast need oxygen to make sterols. If you do a lot of heavy oxygenation during the initial exponential phase, then the yeast can biosynthesize their own sterols. They have the genes to do it, they will do it, they need oxygen to do it, so you've got to aerate a lot to get them to do it. But like I mentioned, the metabolic burden and the overall stress, these are large, complex molecules, and it takes a lot of enzymes and a lot of steps, and it's a metabolic burden. You're better off to feed them this than to make them make it themselves. They can, but what's the consequence going to be in terms of metabolic stress? Okay, just to illustrate, our product, about 3 times the level of sterols of the other leading product. Ergosterol, I showed you the paper that showed that this particular molecule is critical in ethanol resistance and fermentation security, as well as the positive and negative aroma effects. 5 times the amount in our product. We know these things. That's why we blend our products to be enriched in these areas. So, yeast rehydration, survival factors lead to better functioning of yeast metabolism, optimized aromatic intensity, more efficient use of nutrients. We've seen with a reduced GAN environment, with the rehydration, you still get complete ferment with relief of the stress pathways. Lower VA production, less negative sulfur compounds, strong fermentation finish. That's what they're designed to do, and that's, it's a changing paradigm. What it started off with just adding DAP, 40 years ago, 30 years ago, because stuck and sluggish fermentations were a problem, because off aromas were a problem. Still today, many winemakers, they'll monitor your ferments. I mean, you go by, you taste every day, you smell them every day while they're fermenting. You start to smell something a little stinky, what do? Throw some DAP in there, right? And for the most part, sometimes it works, and sometimes, sometimes you may end up generating some heavy sulfides that come back to haunt you later. We've shown through the transcriptomics and through the pantothenic acid versus the DAP additions that it doesn't work alone. DAP alone doesn't relieve the stress, and when you start to smell hydrogen sulfide, there's more going on there than just that. Can you remediate that somewhat? Yes. Are you going to cure the problem? Probably not. Okay, so I mentioned, the changing paradigms. So these rehydration products are not brand new. They've been out on the market for a number of years. But what we've learned through feedback from people as we've introduced these and put them out on the market is that there's really different drivers in white and red fermentations for what these rehydration products bring. In white fermentations, usually you're looking for better aromatics. You still want a good, strong fermentation finish, but the challenges to a strong fermentation finish are not as great as they are in red wines. But the real challenge there is in bringing as good an aromatic component as you can and eliminating off aromas. So we've changed formulations so that we've, you keep the same amount of sterols and fatty acids to make sure you get good fermentation finish up through 14.5, 15% ethanol, but you increase more of the yeast extract guts that lend itself to more of those aromatic characteristics. So that's a new product we call Superstar Blanc, but it's a formulation of a rehydration product specific for white fermentations aromatics. Now, in red fermentations, usually, red musts ferments are done at higher temperatures. There's more phenolics. You encourage extraction of phenolics. Those are detrimental to the yeast metabolism. It's, phenolics bind proteins. That's what they do. What's on the surface of these yeast? Proteins, all their transporters. Phenolics can be inhibitory to yeast. They can make them a little bit unhappy. So it's a more challenging environment. You've got more temperature variation during the fermentation, more challenging environment. So what we found is that if you increase the sterols and fatty acids for red fermentations and keep the level of yeast extract the same as far as just for generating aromas, then you get a product that works really well for red ferments. And that's where we've gone with these products now, is understanding what the demands, at first what the products really relieved in terms of the yeast stress and fermentation, and then realizing that there's differences and trying to make products that address those differences in a focused way. And so just to show, with the Superstar Blanc versus an untreated, you can see that there's a definite increase in the, again, the 4-MMP, 3-MH, 3-MHA. We talked about what those compounds are in terms of flavor, aroma, increase. And then with our Dynastart, which is our the usual generic rehydration product that we developed and put out, and then the reformulated Superstar Blanc, you can see an increase in the aromatics in that. So, the claims we make, it shows up when you actually do the fermentations. Same with the Superstar Rouge. This is the product that's made for red wines. This is it's a product for protection, sterols and fatty acids. Again, I like to make the point, this is not the same thing as a nutrient. Okay? And the point I like to make about this, this is arbitrarily set at 100%, okay, where you're looking at the ergosterol content of these products. What's this guy? How are these guys selling this product as a rehydration product that does these protections and it doesn't even have hardly any ergosterol in it? There's a difference out there on the market. Okay, so protection and nutrition, just kind of the take-home thing from everything we talked about today. Use of cultured yeast can provide many advantages. There's some complexity and diversity things that native ferments can lend, but there's a higher risk of downside. It's not all downside, there's lots of reasons people do native ferments, and more power to them. You've got to monitor your ferments more, you really got to babysit them, make sure things don't go a little sideways. But you get a number of benefits and security from using cultured yeast. And that's kind of where we've gone evolutionarily. And I think a lot of that is driven by, the wine industry has grown tremendously. It's huge volumes and it's larger scale ferments almost across the board. Okay? And that is, if you're a business person, and you better be, or you're not going to be in business for very long. Okay? Risk management. Okay? If you're doing a 200,000 gallon fermentation, the risk of a stuck or sluggish and the remediation you'd have to do versus using a rehydration product and a nutritional product, come on, it's risk management, and it's all about weighing the pluses and minuses in dollars. And that's a good deal of where we've gone with yeast selection, and it's all stepwise with the industry and how it's grown. I think that the quality of wines across the board, even lower tier wines to this day, everything's got to be pretty clean. You got to make a clean wine. Even if it's not super expressive, it's not particularly varietal, if it's not clean, you're going to have a problem putting it out in the market. Okay? And the bar has been raised for wine quality across the board. So the risk gets to be a little too high sometimes. Okay, there's fundamental important difference between protection and nutrition. I'm sure you're tired of me saying that now. Okay, carbon and nitrogen balance is important, but it's certainly not the only nutritional consideration. Complex nutrients are important. Optimal Brix-Yan ratio is changing. Okay, like I said, this goes back to the nitrogen utilization efficiency. What we're seeing is with the rehydration that the nitrogen, you can use lower Yans and still get good fermentation security and good expression. Nitrogen and pantothenic acid work together to relieve stress assimilation and sulfur off aromas. Okay. Proper rehydration increases the yeast viability, ensures strong fermentation, alcohol resistance, more positive, less negatives, those three events. White and red wine must, different challenges, different approaches. So keep your yeast happy, and your yeast will make you happy. Okay, so real quick, Laffort, we're a family-owned company, 1895, I mentioned that. We're the world's largest producer and distributor of enological products. We're in every major winemaking region around the world. We've been on our own as Laffort USA for 5 years in the USA now. Our products were distributed through other folks until 5 years ago. We took it over ourselves. So, we're relatively new, but we're out there in the market with our products. And I can say we're distributed all around the world, sales offices in the major areas and distributors. This is Vines and Vintages here in Ontario. And then, so I always like to close with this quote. I really love this quote as a researcher and a scientist. I like this because you're not always trying to, invent the wheel. The task is not so much to see what no one has yet seen, but to think what nobody has yet thought about that which everybody sees. Who do you think this guy is? Any guesses? This eminent physicist, Erwin Schrödinger, 1933 Nobel Prize winner. Okay, so with that, I'd like to thank everybody for your attention today, and hope you got some good points out of this. And if you'd like to ask any questions, Belinda's going to help facilitate the question and answer session. And so I'll just say again, thank you very much for your attention.
We've got time for one or two questions, ladies and and gentlemen. So, Jen, I'm sure you had one.
I'm okay right now. Thank you.
Anybody else? One question I heard somebody mention earlier on was, do different grape varieties have different types of, or different amounts or different concentrations of the fatty acids, of the C8 and C10 fatty acids?
The grape
Is it down to
varieties?
different grape varieties, or is it more the yeast?
It's more the yeast and what the fermentation condition is, what the environment is, and how it responds to it metabolically. So I guess, it can be in any variety if it's unhappy. If it's a low yan or if there's some stressors in the environment, then it can trigger that response.
Anybody else would like to ask any questions? What about vintage this year coming up? Well, next week it starts, actually.
Well, let me say one thing about varieties. Chardonnay is always a bit of a problem.
Yeah.
And I would say that would be a higher risk for the C8, C10, just because, getting through fermentation in Chardonnay seems to be a bit more challenging than other white varietals.
And then, of course, that causes problem for malolactic, then presumably second ferment as well, if you do, secondary ferment. Any other questions? Esther, would you like to? No? Okay, Peter, thank you very much for your time today.
Thank you.
And I think we've all got Peter's contact details if we need anything in the next few weeks. Thank you.
Thank you.